Hi, everyone, and welcome. My name is Meredith Cagle. I'm the Senior Director for Patient Engagement here at Global Genes, and we're pleased to bring you today a webinar on Batten disease. We have a really full agenda today on the call, some fantastic information, and I hope you're all around to stick around till the end. As Ashley mentioned, should you have to jump off for any part of the time, we will be making this recording available afterwards or to share with anyone who didn't participate. I do want to mention that briefly who Global Genes are. We are a nonprofit advocacy organization whose purpose is to connect, empower, and inspire the rare community. And I hope that through this presentation today, you will find that you feel more connected, empowered with the information, and inspired to take some action. So our agenda today, you just got the welcome from me here. We're going to go right into, after me, a clinical overview of Batten disease with Dr. Emily De Los Reyes with the Nationwide Children's Hospital, Craig Benson and Mary Beth Kaiser from Beyond Batten Disease Foundation, and Noreen Murphy from Batten Disease Support Research Association will be giving us an overview of current efforts in Batten disease research. Dr. Katrin Meyer from Nationwide Children's Hospital as well will be talking about efforts in gene therapy. Maria Graham is a parent here to give us a perspective on clinical trial participation. And Jane Gershwitz from Amicus Therapeutics will be providing an overview on Amicus and their advocacy program. And we would like to acknowledge the support of Amicus in making this webinar possible for Global Genes to bring to the Batten community. We'll open it to questions and answers at the end as time allows, and then I'll just do a very quick wrap. So with that, I'd like to turn the phone and panelist controls over to Dr. De Los Reyes.
Hi, good afternoon. I am Dr. Emily De Los Reyes. I am the Director of the Batten Disease Center of Excellence, and we have currently 4 clinical trials for Batten disease, 2 of which are for enzyme replacement therapy for CLN2, a gene therapy for CLN6, and most recently for CLN3. And my objectives today are to discuss diagnosis and current classification and to review some treatment and future modes of therapy for Batten disease. And these are my grants and consultancy. So Batten disease is the most common cause of clinical dementia in children and adolescents. So previously normal children may suddenly lose their ability to talk or walk, or they may present actually with just language delay, or some of the families may notice that the children are not functioning as well as other children, and they may be diagnosed with cognitive or learning disability. In the case of CLN3 or juvenile Batten disease, most of the time the children may present with visual loss or just problems with their vision, especially in the dark. Batten's disease is a lysosomal storage disorder, and I will explain that further. It is usually an autosomal recessive condition, so that 2 family members may have the genetic abnormality. Rarely there's a form of adult Batten disease called Cuffs B disease where it's autosomal dominant. In autosomal recessive disorders, 2 family members will have the genetic abnormality. More and more, we are finding more molecular mutations throughout the world, especially with advances of genetic engineering and exome sequencing. There are currently 14 presumed genes. Because of the lysosomal disorder, there's accumulation of lipopigments called lipofuscin in the cells. So again, the overall presentation of these children may be language delay or language regression. In CLN3, as I mentioned earlier, the children may present with progressive loss of vision. Epilepsy is very common in children with Batten disease and may be extremely difficult to control, so that some of them may present just febrile seizures and begin to have partial or generalized seizures, and as the disease progresses, they may have worsening of their seizures called myoclonic and atonic seizures. And currently at that time, that may be where the doctors may try to diagnose with more expensive genetic testing, and that's where they find the Battens. It is our task. right now to find the children earlier so we could start treatment earlier. As I mentioned earlier, they can have cognitive decline. They can also have motor regression. And one symptom that may be underdiagnosed are sleep disorders. So this is a cartoon that I borrowed from Dr. Bustani of the lysosome. You can see the lysosome in the middle of the cell. It is, I call it the vacuum cleaner of the cell, so it helps break down all the unnecessary proteins or lipids, and without this function, this vacuum cleaner does not help with, is unable to clear the cell of all this unnecessary objects. And as I mentioned earlier, that's where you have storage disorders called lipofuscin. And there's a question as to whether that's the cell trying to rescue itself. So as I mentioned earlier, the lysosome is very important in degrading and taking up protein. It has a significant role in macro and micro autophagy, so a lot of the experimental drugs may have actions in that. The lysosome has associated membrane proteins and may facilitate uptake and digestion. So the loss of this normal function to degrade this complex substrates may cause problems with degradation, and this may cause toxicity to the cells, the tissues, and organs, especially the brain. So this is the current classification. We classify it by the number. And I will show you some more slides. So there's 14 types of Batten's disease, and we have identified mutations in 13 of the 14. So in the old days, we classified it by at the age of presentation. So we would say CLN1 is infantile NCL, CLN2 is late infantile, and CLN3 is juvenile. With the advances of genetic technology, we now know that CLN1 doesn't just present early on. We have children who present later on. And the same way with CLN2 or CLN5, 6, 7, 8, all those different CLNs may present anywhere from late childhood or even to adulthood. CLN3 remains very fairly stable, so that it may present usually between 4 to 8 years of age with visual loss. And in CLN3, we know that there's a transmembrane protein, as opposed to CLN1 and 2, where we know that there are specific substrates that are missing, and with the hope in CLN2 to replace that to attenuate progression of disease. As I mentioned, in the old days, it's age of onset, and we used to perform skin biopsies on the children and biopsies of their conjunctiva to look for ultrastructural characteristics like deposits like curvilinear bodies and fingerprint bodies. So these are examples of the different curvilinear and fingerprint bodies. For in the Batten's Disease Center of Excellence, we promote a multidisciplinary treatment, not just one doctor, but a combination, a team. We call a team to take care of these children. So we can have not just a pediatric neurologist. We have a physical therapist, a speech or feeding therapist, and occupational therapist. I call this. I tell my families that these children are athletes. We have to make sure they have good nutrition, that we have to keep them well hydrated, and that they remain strong. We also have ophthalmologists to help with their vision if we could help them. And we employ nurses to help them with various resources, especially the palliative care teams. And then, of course, GI or nutrition to keep them with their nutrition, because we think that's very important. In the old days, we used to just say to the families that we would like to maintain their quality of life by improving seizures, because seizures are so difficult to control. Although with the treatment now, it's much easier. These children may present with myoclonic seizures, partial or generalized tonic-clonic seizures. The most common medications we like for these children are more of the drugs that are helpful for what we call generalized seizures, like zonisamide, valproic acid, and clobazam. Although sometimes they may worsen with some medications like that are partial onset, but some children also do well, so very small population may do well. Some of the children may present spasticity, so it's extremely important to keep them stretched and keep them with physical therapy so that they can continue to walk. When the children develop swallowing problems, they may not feed as well, so it's very important to monitor for choking, and sometimes we thicken the feeds. Later on, if the families feel that the children are just too exhausted to eat, we may even recommend G-tubes. Nowadays, in wintertime, these children are extremely important to vaccinate the children because every time they get sick, they slide or lose their milestones. So I always advocate for influenza vaccine and all the rest of the immunizations. I think that's extremely important because once they get sick, it takes the life out of them and it worsens the disease. As I mentioned, I do not see any reason I may space out the immunization, but I emphasize immunizations are very important, and good weight gain and good nutrition is very important. So as the disease progresses, I borrowed this slide from Dr. Williams, who published this study alongside with multiple other co-authors looking at quality and the importance of quality of life for these children, not just for the children, but also for the siblings, and the parents are very important. I talked about medical management and family support. In some of the other cases where we still don't have treatment, they may it is extremely important to involve palliative and hospice care. Lately, we know that with the involvement of gene therapy, we know that the quest to find the cure is ongoing, and Nationwide Children's Hospital is very committed to helping these families. And again, this slide just illustrates how it is very hard to find a drug that will target the brain completely, but and even traverse the blood-brain barrier. So currently, as we know of, we have one drug that's approved in the market for CLN2, and that's cerliponase alpha for children with transpeptidal peptidase deficiency, or CLN2, or as commonly known as late infantile. There was previously intracerebral gene therapy through Weill Cornell, and currently there are clinical trials for CLN3 and CLN6 disease. And I'm sure the other the other speakers for this will talk about bone marrow transplant and stem cell. And again, the quest for other less invasive, like neuroprotective agents, is ongoing also. And I think looking for seizure control is also important. Thank you very much.
Thank you, Dr. Davis. That was wonderful. I appreciate that. We're going to go ahead and transition now to Mary Beth Kaiser with Beyond Batten Disease Foundation.
Thank you, Meredith. And thanks to Global Genes for inviting us to participate on this webinar. I also wanted to share our appreciation for all the organizations on the call for the good work they are doing in this field. My name is Mary Beth Kaiser. I'm the president and CEO of Beyond Batten Disease Foundation. And I'm here with Craig Benson, who is the founder and board chair, and more importantly, is the dad of Christian, who has juvenile Batten disease. I want to give you a little background first on our organization. Beyond Batten is a nonprofit public charity with a mission to raise awareness and funds for research for Batten disease, and more specifically for the CLN3 or juvenile form of the disease. The foundation was established in Austin, Texas after the Bensons received Christian's diagnosis when she was 5 years old. And today we work with families all over the world to advance the science and fund potential treatments for juvenile Batten disease. My slide's not moving for some, there we go. Over the last 10 years, we have executed a unique research strategy which focuses on partnerships with other Batten families and foundations, as well as other neurodegenerative disease groups, in order to pool resources to have the biggest impact. Our most important partners are the affected families that we work with. BBDF provides a nonprofit umbrella and scientific leadership for Batten families and their communities who are interested in raising funds for research. This is a sampling of the directed funds who we work with very closely. In addition to these funds, many other families and their communities participate in fundraising activities and are part of our patient advocacy network. And we also work with hundreds of other organizations to advance the science of CLN3 disease. These are just a few, some you may recognize. All of that partnering has allowed us to invest over $26 million in research over the last 10 years. And that money has come from direct donations as well as co-funding projects with other like-minded organizations, like some of the ones you saw on the previous slide. The amount of investments has allowed us to have a prioritized research portfolio where we have been able to fund many projects from basic academic science to preclinical development. Our goal has always been to put research dollars where they are needed most and to support and advance as many projects as possible. We don't care where the treatment comes from. We just want one as quickly as possible. So early on, we were able to fund a large number of projects. In the last few years, as lead candidates for treatment have emerged, we have narrowed our focus on more preclinical development while continuing to support ongoing research programs. In order to give you a scope of our work, we wanted to share some of the highlights and accomplishments from our research program. And I know this is a lot of information, so I'll just touch on it quickly. The Foundation's first investment was a rare disease genetic test to detect Batten and over 800 rare and medically devastating diseases. And we've made significant investments in other areas, including the discovery of drug targets in medicine, which means identifying which DNA, protein, or other cellular targets can be manipulated to inhibit disease. One of the ways we did this was by bringing together over 55 world-renowned researchers for a two-day conference at the New York Academy of Sciences to explore drug targeting the lysosome. We've also invested in screening drugs in an effort to repurpose drugs that are currently available and might have an effect in CLN3 disease. And our strategy includes incorporating cutting-edge methods and platform technologies to give the research community the tools they need to advance the science. This includes the development of a first-ever commercially available Batten iPS stem cell line so that researchers can do their testing in actual Batten brain cell models. We're also interested in advancing any and all new emerging therapies, and we've invested where there has been a need in projects like gene therapy and exon skipping. And as a patient advocacy group, we appreciate the importance of natural history studies and registries. And to that end, we have supported the NIH's Natural History Study and funded the work of DemChild to connect CLN3 registries worldwide. And we also work very closely with leading clinicians, key opinion leaders, and regulatory consultants to determine the best ways to measure effectiveness of treatments, which will be beneficial for any and all clinical trials. Sorry, my slide's slow to move, but there we go. So in addition to the program highlights I just went through, BBDF funding led to the discovery of a combination therapy that is shown to slow the progression in Batten disease models. And we've spent a great deal of time over the last few years on the preclinical development of this combination therapy, including engaging regulatory consultants to help us navigate the FDA process for clinical trials. In May of this year, we had a meeting, our pre-IND meeting with the FDA, resulting in some direct feedback and guidance for the program. And we were especially pleased that the agency communicated their understanding of the need for the therapies for this population, and they also communicated their willingness to work with us to help move the program through the process as quickly as possible. This picture includes the team that attended the meeting on our behalf, and there was a lot of brain power in that room. So as I mentioned, the FDA provided a clear path forward for the therapy, which we are calling BBDF 101. And to illustrate our next steps, I wanted to show it against the traditional FDA approval process, which is the line at the top. Typically, it's multiple phases and steps for new drug approval. And the short story is the FDA said that following one short rat toxicity study, that we're going to be allowed to go directly into a phase 3 trial. And in addition to that, our plan also includes expanded access and compassionate use for kids over 18 with CLN3 disease. So today we're targeting the late spring 2019 to begin the clinical trial, and that's our best guess at this point. We're working very hard and moving things along as quickly as possible. This treatment is very exciting for the juvenile Batten disease community, and we think it also could be impactful for other neurodegenerative and lysosomal storage diseases. So we're anxious to see how this can treat Batten and treat beyond Batten as well. So, this is a summary of our activities over the past 10 years and the direction we're headed over the next few, and that's a little hard to cram into 7 minutes. We are definitely available to answer any of your questions or to talk more about our activities anytime. So we appreciate your time today and the opportunity to share what we're doing, and we appreciate getting to work with so many of you on these projects.
Thank you so much, Mary Beth, and I appreciate you getting it in there within your time. I want to introduce now Noreen Murphy from BDSRA.
Hi, thank you. I am the Patient and Family Education Coordinator at Batten Disease Support and Research Association. We're really excited to be a part of this webinar and grateful for Global Genes for hosting. So for those of you who are not familiar with BDSRA, our mission is to support Batten families, fund and facilitate research, and advocate for action. For over 30 years, BDSRA has served families of children with all forms of Batten disease. A full complement of support services is available to families, including a large family conference held each summer, a vibrant online community for support and education, grants for equipment needs, and more. BDSRA's research program has funded registries, animal models, biospecimen collection, and other major building blocks important to the Batten scientific and drug development process. With many others in the rare disease community, like Global Genes, we advocate for a brighter future for all by building coalitions, educating decision makers, and representing the important patient voice in the scientific community and everywhere possible. Just wanted to dive a little deeper into some of the ways that BDSRA supports families. First, we host an annual 3-day family conference. This past year, we had about 480 attendees. These include all members of families, parents, affected children, siblings, and extended family members, researchers, clinicians, and members of industry. The focus of the conference is to connect families with one another and experts in the Batten community. We have skilled nursing care for affected children and a comprehensive sibling program that goes over the course of the 3 days. We have a closed Facebook page for parents and caregivers. This is a really great resource. We have about 1,000 participants, which includes parents of those with children who are living with Batten disease and bereaved parents. This is a great way for the real Batten experts, the parents, to share advice, ask questions, and when necessary, to vent to people who understand what they're going through. On this closed page, we host Ask an Expert chats that cover a variety of topics, including seizures, behavioral problems, school issues, grief, and many others. We have a monthly newsletter for sharing updates and news. Our staff is always available by phone and email to help families find community resources and provide support. We're happy to answer any questions, provide a listening ear, and connect families to one another for peer support. BDSRA also represents the patient voice wherever and whenever we can. This includes conferences all over the world, conventions, and meetings with the FDA and NIH. So this is a really exciting time for the Batten disease community. I think research seems to be progressing faster than ever. This quick chart outlines the companies and organizations that we know of that have announced their intention to work on potential therapies in Batten disease. As you can see, there's a lot being done in a lot of the forums. At the recent NCL Scientific Conference, which is held every 2 years, there were presentations on early work being done in even more forums, so I expect this chart will grow quickly. I do have one small correction to make, though. Preclinical Therapeutics, which I have listed as working in CLN 2 and CLN 3, I've listed as working in CLN 1, but should be listed as CLN 3, sorry. So I've asked each of these companies to provide either a slide or some general information about their programs for me to share. The slides are very basic, but I've also asked them to include a way for families to contact them for more information. Don't have a ton of time, so I'm just going to touch on them very quickly. Mary Beth already talked about their program, so I will start with Abeona Therapeutics. Abeona is working on a gene therapy programs in CLN 1 and CLN 3 disease. They have outlined each of their programs here, but I just wanted to mention that in both of them, they are targeting initiation of phase 1/2 trials with the primary objective of evaluating safety in 2019. If you have questions about their programs, you can email Judy or Michelle. Next up, we have Amicus Therapeutics. Amicus is working on gene therapy programs in CLN 3, CLN 6, and CLN 8. Jane will actually be speaking later more specifically on their work, so I'll just leave it at that. Next, we have the Foundation for Batten Hope. They're working on gene therapy program in CLN 7. They're currently working on toxicology study, viral vector development, and initial animal studies. Their next step is recruiting for a natural history study with UT Southwestern. This natural history study recruitment will include CLN 7 and CLN 5 patients. Feel free to reach out to Gina with questions. Then we have Circumvent Pharmaceuticals, who is working in a number of forms, CLN 1, CLN 3, CLN 10, and the adult form of Batten disease. Their program is a small molecule therapy. They have completed key toxicology and preclinical and are developing a clinical formulation for IND-enabling studies. We have Polaris Therapeutics, who is working in CLN 2 and CLN 3. This is also a small molecule program. They expect to file their IND in the second quarter of 2019 and start their clinical trials in the third quarter. Next up is REGENXBIO, who is working on gene therapy in CLN 2 disease. Their program is currently in the preclinical stage, and they are making progress towards submitting an IND in 2019. They are recently granted orphan drug status by the FDA, and you can reach out to their wonderful patient advocacy team at the address listed below. And then lastly, we have Spark Therapeutics. Spark is also working on a CLN 2 gene therapy program. They are in the preclinical stage of development. You can read sort of through their program information there and reach out to them at that address listed. I know that was like a ton of information in just a few minutes, but I'd be happy if you want to reach out to sort of recap any of this. We're excited to be working with all of these organizations on different programs coming up, and we're excited for all the work that's being done by everyone else on the webinar. Thank you.
Thanks so much, Noreen. And so, by the way, we do have a couple questions coming. I just wanted to, so that we stay on target, we will be answering Q&A at the end. So thank you for your patience on that. I'd now like to introduce Dr. Catherine Meyer, who's the principal investigator at the Center for Gene Therapy, to speak about gene therapy programs. And Dr. Meyer will help you out with your slides. Just give us the next slide, go ahead, and we'll move it forward for you.
Okay, thank you very much. I'm very happy to be on the call. As mentioned, I'm an assistant professor at The Ohio State University and principal investigator at the Center for Gene Therapy at Nationwide Children's Hospital in Columbus, Ohio. And yeah, I will probably need some assisting. So please, next slide. I have disclosures to make. I'm a consultant for Amicus Therapeutics, and I'm going to talk about preclinical proof of concept data from studies that we've been performed for treatment of patients with Batten disease. Next slide, please. So we're using adeno-associated viral vector 9, AAV9, which is a natural occurring virus that can infect humans and other species, but it doesn't cause any disease. And it is a virus that has been used very successfully for other diseases, including spinal muscular atrophy, Duchenne muscular dystrophy, with multiple different types of delivery routes. And it really has been developed at Nationwide Children's for the applications in these neurodegenerative diseases. So gene replacement therapy, just briefly for maybe the families out there that are not as familiar with it, it's pretty much like if you have a defective gene, that means you have a car in your garage that isn't working. So what we're trying to do with the gene replacement therapy is to put in a second car that is actually working. And while the non-working car is still there, you can now use the new car and you can still get around and you can circumvent the problems arising from the first car not working. This approach has been very successful and is moving forward into clinical trials, or already has been tested in patients in multiple applications, including spinal muscular atrophy, Rett syndrome, ALS, CLN6 and CLN3, Batten disease at different stages of the development. And it's really an advantage to use the same viral vector because we have a very long-lasting experience for both dosing as well as safety studies. Next slide, please. So here's an example for a study that we've done in CLN6 mice. On the very left side, you see healthy animals. So you all know that in Batten disease, one of the major hallmarks is the accumulation of this autofluorescent storage material that just cannot be cleared out and leads to death of neurons. And so you can see that very well in the middle panel in an animal model for CLN6 disease, where we have accumulation of this green material, which we can also call trash, throughout the brain. And when we treat these animals with our viral vector that delivers a healthy copy of the CLN6 gene, you can see a very strong reduction of the accumulation of the storage material, and the quantification is shown on the right. Next slide, please. When we successfully clear the accumulation or prevent the accumulation of the storage material, you can also see that we can reduce the inflammation that happens in the brain. So again, on the left side, you see a healthy mouse. You see very little accumulation of staining for astrocyte activation as well as microglial activation, which are two cell types that are very important in the inflammatory response in the brain. Untreated CLN6 mice in the middle have a very strong signal, and animals that were treated with our viral vector have a very strong reduction again. Next slide, please. So when we are able to prevent the accumulation of the storage material, we also saw a very strong improvement of the mouse behavior. So this is basically, these are tests where we put our mice through a sports event or an Olympic event, basically, where they have to show how good can they walk on a spinning wheel, how fast can they swim, and are they able to detect a hidden platform in the water using visual clues. And as you can see, in the dark blue, untreated CLN6 animals that carry a mutation start having significant difficulties at 8 months of age, and to the point that at 12 months of age, they're basically not able to perform this test anymore. Whereas the CLN6 animals that got our gene therapy treatment, which are in light blue, are very similar to the wild type animals, and they are able to run on a spinning wheel on the top left. They are able to swim equally fast compared to the wild type animals on the top right. They are still able to find a hidden platform in a water maze, although they do see that they do get additional difficulties at the later time points at 18 and 24 months of age. And there's additional benefits in just the gait and the way the animals walk, which is shown on the bottom left. Next slide, please. Also, very importantly, the treatment had a very significant effect on survival of the animals. So untreated animals with the CLN6 mutation die at 14 months of age. It's a very steep drop in survival. And when you look at the CLN6 animals that were treated, their median survival was 21.5 months, which is not statistically significantly different from the wild-type mice. So we had a very clear increase and benefit in survival in the CLN6 mice with our treatment. Next slide, please. As a next step, we did dosing studies in non-human primates just to make sure that we get a good expression throughout the brain and spinal cord of our transgene. And this slide just shows you various regions of both brain and spinal cord that show increased expression of the CLN6 protein when the non-human primates were treated with the CLN6 gene therapy. Next slide, please. Next slide, please. Okay, so now I'm switching to another program we've been working on using the exactly same strategy, replacing or bringing in a healthy copy of the CLN3 gene and using the CLN3 mouse model that carries the most frequent mutation that is found in human patients. And very similar to what we've observed with CLN6, again, we saw a very strong effect on storage clearance, both at early time points, as early as 2 months, as well as later time points of the disease, up to 8 months and even later on. So again, we have very robust prevention of the storage clearance with our gene therapy product. Next slide, please. Thank you. So this is again similar to the CLN6 program. We again were able to strongly reduce the microglia activation and the astrocyte activation, which are markers for inflammation. You can see in the middle panel, the untreated CLN3 mice have very angry cells that are ready to kill, whereas the wild type and the treated animals have much less inflammation. Next slide. Although the CLN3 animal has a very mild phenotype, we were able to see differences in the behavior, again, very similar to what we've seen in the CLN6 animal models, where the treated animals, the animals that got our gene therapy treatment, were much more similar to the wild type animals compared to untreated CLN3 animals with the mutation. Next slide. In terms of survival, as I just mentioned, the CLN3 mouse model doesn't really have any deficiency in survival. So at this point, all the animals, wild type, untreated CLN3 animals, as well as 89 treated animals, are still doing well in terms of survival. So there's really no difference, but we also didn't expect it at this point. Next slide. As previously, we also, again, tested the expression of our transgene in non-human primates, which we really believe is very important to confirm before we take the next step, get into human patients. And again, we've seen very widespread expression. Here we're looking at RNA levels, not protein, because the antibodies are not very good for CLNC protein. We can see very widespread expression of the transgene throughout the brain, including retina as well. Next slide. Next slide, please. So being really encouraged by the data we gathered from the CLN6 and the CLN3 projects, we also moved forward with an additional project for CLN8 Batten disease, again, doing exactly the same thing, trying to bring in a healthy copy of the CLN8 protein. We were very encouraged to see, again, the same effect on accumulation of the autofluorescent storage material in the middle or untreated mice, left and right, or wild-type mice, and also treated CLN8 animals. Again, we had a very strong reduction in the accumulation of the storage material. Next slide. Same here. Next slide, please. Thank you. Again, we had a very strong reduction in inflammation in both cell types, astrocytes and microglia, which is very important to support the neurons and throughout the nervous system. Next slide. Again, here this mouse model has a stronger disease phenotype compared to the CLN3 mouse. So the mouse Olympics are much clearer, Again, on the top left, we have the ability to run on a spinning wheel. And as you can see, the untreated CLN8 mice were not able to perform this task anymore at 10 months of age, whereas there was no difference between the wild-type animals and the treated CLN8 animals. Very similar observations when we're looking at pole climbing tests, where we look at how long does it take the mice to climb down a vertical pole. If we put them on the very top, that's the bottom left panel here. The CLN8 animals really take a very long time to climb down, while the treated animals are very similar to the wild-type animals. And again, when we put the animals for swimming, the swim speed was very similar to wild-type animals, even at this late time point at 10 months of age. And they also were much better in finding a hidden platform in the water using visual clues, although we had a significant difference there too compared to wild-type. Next slide. Here you can see the survival curve. At this point, none of the untreated CLN8 animals are still alive, while both wild-type and the CLN8 treated animals are doing very well. All of them are alive and looking really good. Next slide. Maybe some of you have heard that we have initial encouraging data from the CLN6 clinical trial where we have dosed patients with this vector. Here you see the data from the first 2 patients that had a very similar onset of symptoms, but one sibling was treated at an earlier time point and is doing really well, as you can see in the green line. We're looking here at the Hamburg scale for motor and language abilities combined. So this is very encouraging, still early. We have to analyze the data more, but the first step towards clinical application has been done, and we're very excited to move forward. Next slide. So from the CLN6 clinical trial data, we can just a quick overview. This is a single-arm study. All of the patients received the gene therapy. 10 patients were treated so far. Additional patients are going to be included. Generally, the treatment was very well tolerated. We have encouraging preliminary efficacy data, and Amicus is expecting to release additional data in 2019. Next slide. The average follow-up duration is 12 months at this point. We'll follow the patients up to 24 months. There have been adverse events, which happens in all the clinical trials. Everything has to be reported. Most of them were minor and mild and unrelated to the treatment. A few of them might have been severe, reported in 4 patients, and none of them has been a grade 4 or a grade 5 to date. So what we saw was a T-cell response and antibody elevations that were not associated with any clinical manifestations, which is something that we had expected, and no change in treatment was required. The DSMB board has permitted the study to proceed and enroll additional patients. With that, that's all I want to present today. We're very excited to be part of this community, and we've had a lot of support from multiple different Batten disease organizations, including Charlotte and Gwyneth Gray Foundation, 4 Batten Foundation, 5 Batten Foundation, and we're very excited to move these therapies towards clinical trials. Thank you.
Thank you so much, Dr. Meyer. I appreciate that presentation. We're now going to go ahead and Maria, we're going to give you panelist control here. Well, actually, we're not going to give you panelists. We're just going to give you audio control. Maria, as a mother of a lovely daughter, Layla, we want to hear about her personal experience with Batten disease and clinical studies. Maria.
Hi, everyone. Good afternoon. I am the mother to Layla, who is now 8 years old, and she has CLN2 late infantile Batten disease. Layla got her diagnosis of Batten disease when she was 4.5 years old. The reason why we got the diagnosis is because she started having seizures 1 year prior to that, and her seizures progressively got worse. She had a small speech delay in the beginning, and I noticed some regression of her speech and her gait. And so they did genetic testing, and we found out that she had Batten disease. We are from Atlanta, so we were at Emory Genetics, and they came into the room and told me that my daughter was going to die and pretty much just go home and spend as much time with her as I could. About 3 weeks, this was in the first week of October in 2014. About 3 weeks later, the geneticist called me back and told me that there was a clinical trial that was about to start at Nationwide Children's Hospital in Columbus, Ohio. And so he put us in touch with Dr. De Los Reyes, and we found out that there was a clinical trial starting that would deliver a synthetic enzyme into Layla's brain to replace the enzyme that she was missing. We were very nervous, apprehensive. My husband first was the one to speak with Dr. De Los Reyes and did not really understand what she was saying. We were scared that she was going to be in pain. We didn't know if it was worth the risk. But the only other option was death, so we decided to become a part of the trial. We picked up our whole family, and we moved from Columbus, Ohio we moved to Columbus, Ohio from Atlanta, Georgia, and we are still here. We have been in this clinical trial now for 4 years, almost to the day, and it's been a roller coaster, but it was definitely worth it. I was just going to note that my daughter was born completely normal except for a mild speech delay. She started getting the treatment at the 1st of December, and she was going to turn 5 years old on March 15th of 2015. Within the first 3 months of the treatment, my daughter had severe regression. She lost the ability to walk, to talk, and then soon after her 5th birthday, she got extremely sick and was in the hospital for 2 weeks. She kept getting a virus upon virus, and after that, she was regressed to the point of being like a rag doll. She could not eat. They put a G-tube in, and we continued with the trial because we had said we were going to give it 6 months. So we continued with the trial, and she now is back to sitting up on her own. She can stand with assistance. She can walk with assistance. So this has been a great positive experience for us. If you look on the pictures, this is my daughter. All 4 of these pictures are of my daughter after treatment. The one on the left is the youngest, and then the third one is soon after that. But then these last 2 pictures are very recent. My daughter would probably, in my heart of hearts, I know my daughter would already be dead without this treatment. I will say it was a little easier for us to sign on to this clinical trial because even though my daughter was the second child in the United States to receive this treatment, which is Brineura, and it's the CLN2 treatment that has already been FDA approved. The reason why it was easy for us to say yes is because there was already children that were doing this for, I'm thinking, maybe 9 months before my daughter started in Europe, which in Germany and England. And so we knew that, so far it was pretty safe. So I will say that it was an easier decision for us to try it. But I would say, with all parents of Batten disease, the inevitable of no treatment is the worst possible outcome. So why you wouldn't want to try some kind of treatment, no matter if you already knew it was safe or not, is, to me, I just feel like there's no option, because you want to keep your child with you for as long as you possibly could. And my daughter, I feel, has a good quality of life. She is very happy. She goes to school. She loves school. She loves watching her movies. But it has been hard because she has lost her vision, and we have to go and spend one night in the hospital every 2 weeks. And she absolutely hates the hospital. So that has been hard on her, but it is relatively simple, her infusion. And, it's just kind of like most of the time we're just kind of sitting in the hospital. She goes through about 20 minutes of angst in the mornings to get the infusion started. But other than that, it's not too bad. It's been very hard being away from my family for 4 years. We didn't know anyone when we moved up here. But I will say that the BDSRA and the drug company BioMarin has definitely made this transition smoother. The drug company has completely taken care of us, as in like helping us financially. They give the drug for free. The BDSRA has implemented all of that by getting us set up with a place to live and, keeping us taken care of. So there are good points and bad points, but the good far outweighs the bad, especially since my daughter is about to be 9, and she is still alive, and she is still smiling, and she is still verbalizing, and she can do things that most CLN2 children with non-treated CLN2 children that are 5 years old could not do. So we are very, very grateful, and yeah, that's about it. That's our experience.
Thank you so much, Maria, for sharing that personal experience with everyone. And now I'd like to welcome Jane Gershkowitz with Amicus Therapeutics to tell about their current efforts and advocacy program.
Thank you very much. Thank you to Global Genes for coordinating today's webinar, and to all of the speakers who shared some really amazing information. And of course, thank you to the well over 80 people who are listening in to this webinar. I think it's great, and it just speaks to the importance of sharing information and learning together as a community. It also points to something that was said earlier, and that, just trying to advance my slides, excuse me. It goes to the point of the interest that there is between academic and industry researchers, and that it has come a long way from years ago. And just so you folks know, I've been working in rare lysosomal storage disorders for over 20 years. So to see this advancement and this increased interest is just wonderful. And truly, I, both personally and professionally, and all of my colleagues at Amicus, we're really honored to extend our commitment through our recent entry to the Batten disease community. Our slides are not advancing, but in the interest of time, maybe the folks there at Global G, there we go. So I think we'll have, if we still have problems, we're going to have you guys advance if you don't mind. So just briefly, from the very beginning days of the company, we have had our belief statement, and it's a belief statement that was penned by our employees. And our CEO, John Crowley, essentially said to then a group of around 25 or 30 people, please come up with what you think is our corporate belief statement, which will be the anchor of what we do and how we do it. And the only 2 words that they were given were, we believe. And this is one of the statements that has come back and that has, as I said, remained an anchor for us. And we're committed to improving the lives of patients and families affected by rare diseases And as I said, we're really honored to now include the Batten community in that commitment. Just briefly, our history, I'm not going to go into this, it's a little bit of an eye chart for sure, but just wanted to show you that from the earliest days of the company, which go back to 2002, which for some of us is not that far in the past, we really have expanded our work in rare diseases, particularly in lysosomal storage disorders. And it was just in the fall of this year that we were able to really enter gene therapy, which was something that well over a year ago we decided was something that we wanted to do, because what Amicus does is we look at where is there the greatest unmet need in a rare disease, and then what might be the technologies, the platform technology or plural technologies that could be brought to bear to fight that unmet need. And so we're very pleased with the acquisition in September of Selenex and working on a range of Batten subtypes, as well as our collaboration with UPenn, which advances us into a total of about 14 pipeline programs. And we are a global organization, so this just shows you where we're based in terms of our headquarters here in the States and internationally in the UK, but where we have offices, and then we have presence in terms of boots on the ground, as we say, around the world. And we're currently sitting at well over 500 employees who really are committed to rare and orphan disease. And how we measure our success is something that we talked about for a long time here at Amicus. Certainly we are a public company and we have an obligation to our shareholders, but above all, we believe we have an obligation to the patients. And so we measure success by how many individuals are being touched by an Amicus product, be that an investigational drug within clinical trials or a commercially approved medicine. And our goal is to be at over 5,000 touch points by the year 2023, and I would say that we are on our way there. So talking about community, rare disease community, which is all of us, there are many stakeholders, and to those of you living with Batten disease, working on Batten disease, we consider you certainly part of the community of us. And this is really something that we have developed through our patient and professional advocacy team here at Amicus, and we'll tell you a little bit more about that toward the end. But it's looking at all the different diseases that are represented that we work on. And as I said, the different stakeholders, the different partners who come together to make this community of us. So we're really driven by the odyssey that people have when they live with a rare disease, the experiences, their involvement, certainly the relationships that we develop and that we nurture, and the outcomes of people living with rare disease. That's what drives us every day. And gaining that understanding of what's happening with the patient community, whether it's individual families, the patient advocacy organizations, as well as the physicians and other healthcare providers that help to support those individuals. We try to understand as much as we possibly can. The one thing that we say here, and I have been saying over and over again over the years, is we can only try to understand the patient experience. We can only try to understand the disease. Unless one lives with the disease themselves, they can't say that they know it. And we certainly appreciate and are humbled by the experiences that all of you have in living with disease and your willingness to share and teach us. And we learn, obviously, through those relationships and working collaboratively. And the whole point is to come up with meaningful outcomes for people across all of these stakeholders. In learning from all of you, we are a little bit off. We're missing a slide here, or maybe we could go back to slide 10 for a moment. Try that. Okay, we're missing a slide, but no, that's okay. So we actually say, as part of community of us, ask us. Ask us about what it is that we can do to help you. We know that the patient advocacy organizations are critical in helping people make the connections, share their experiences, certainly reduce the isolation that can come from living with the rare disease. And we learn from all of you, and in turn, as I was saying, develop that understanding, but also develop an understanding of what might be helpful, what kind of information might be helpful, so that people can become their own best advocates. And that's what this and the next slide will refer to. You can see here from our work in Fevre and Pompeii that, thank you, that we've developed what we consider some simplified disease awareness tools, infographics, and from there then go into some more specific signs and symptom-related materials. And we currently are in the process of developing infographics for both CLM6 and 3, and also for CDKL5, which is another rare neurological disease that we currently are working on. And besides disease-specific materials, we are working on providing general rare disease materials. And so you can see here, and this can come from the need that people say when we don't understand the clinical trial process, or can you help us better understand what is informed consent? And we know that the many different study sites that we work with are working to make sure that people understand that, those types of issues. And so what we have done as a result of what people are asking for is provide these non-disease specific, just general information to help people understand research, how to read clinical research publication, for example, what's important about compliance and adherence in medication, the informed consent process, the drug development process, et cetera. And so these are available to individuals, to organizations, as well as to healthcare professionals and study sites and treatment centers. So besides asking us, tell us, and we have a range of events and activities where we really learn from people living with diseases, and you can see here, and this may not be very different from what some other companies do, but we think that the approach that we take might be a little bit different in some cases. And one of those approaches is our patient advisory boards. We have had individual patient advisory boards for every disease program at Amicus since the program began back in 2007. We established it looking at what was or what wasn't already existing in general healthcare and industry, and then we developed a unique approach to these patient advisory boards that really complete the triad with our medical and scientific advisory boards. It's become an institution here at Amicus, and in fact, I'm proud to say that it's an approach that some other biotechs have adopted over the years. By learning from patient advisory boards, by learning from people who come in for lunch and learns, for roundtables, by us attending congresses and patient meetings and conferences and learning, we're then able to develop some initiatives. Thank you. To develop some initiatives that we think make a difference, not only for patients and organizations, but for healthcare professionals and also for Amicus. And one thing that we've been doing over the last several years are surveys. These are surveys to understand both patient and clinician point of view around living with the disease, what are the burdens of disease, potentially unmet needs, looking at similarities, looking at gaps, and from there then we can decide and work together with our partners to figure out how to fill those gaps. Here are just two examples, one from Pompe disease and one from EB, which is epidermal lysis bullosa, a very rare and devastating connective tissue disorder. And what we learned from these is that there's more work to be done in terms of patient experience, disease understanding, that's separate from the clinical trial process. And it's an opportunity to publish. And in this publishing opportunity. It's certainly, you can't see it here, but we're working collaboratively and co-authoring with leaders of patient advocacy organizations, as well as with healthcare providers and our own team members. And this is something that we think helps to advance the capacity of the patient organization, and we certainly look forward to working with the Batten community to determine what kinds of information might be helpful to garner and to publish. And as the previous slides show, what kinds of tools, what kinds of resources might be helpful to the community. We do not develop these things in a vacuum, but it's all based upon that tell us and ask us approach that we take. Next slide, please. So this is something that came certainly from a family that lives with rare disease, in this case with Fabry, and it was something that the family did to just try to instill positivity in the face of living with a rare chronic condition. And I heard the mom present this, what she called our good stuff, at a conference, and later approached her through the patient organization in her country and said, what you're doing is really terrific, and we would like to give this to other people who are living with rare disease to help them understand that as difficult as the challenges might be, there certainly are positive things that happen as well. Family was very happy that we were willing and interested in doing that, and they became consultants on the project. And this is something that we make available to all of the rare disease communities with which we work, and we actually are also putting this forward with physicians and treatment centers so that they can help patients think about the positive side of things. And we'd be happy to share this information with folks. And it is cited from literature, the importance of maintaining a positive outlook. This is one of the things that we've put together in the last couple of years from our team. And our team, I'm proud to say, is growing. This photograph is from last summer from an offsite that includes our summer interns, our international team, our global and US teams. Don't expect everybody to be able to copy down emails there, but if you go to the next slide, you can see that there's a way to get in touch with us both here in the US, which is the top email, patientadvocacy@amicusrx.com, as well as our international one. And we would say just get to know us as we try to get to know you. You can start to look for us at some of your conferences in the community, but do not hesitate to reach out to us. The only way we will understand Batten as much as we can is to learn from all of you. So with that, I would say thank you, and I would say join us as part of our community of us. And again, thanks for this opportunity.
Jane, thank you so much. And we will be sending out these slides to everyone who registered for the webinar, so you'll have access to those email addresses to follow up with Jane and her team at Amicus. So at this point, I'd like to open. We have some questions that came in, and I'm going to ask them, and if anyone on the panel, if you're able to answer, if it applies to you, we're going to open your audio now. Are you good?
I'm going to unmute them.
Okay,
If the panel, we're going to unmute the whole panel. If you could mute yourself on your end, if you don't have anything to include from the questions, please just mute yourself so that we don't have as much back noise for our audience. So again, everyone is becoming unmuted from the panelists. Again, you can mute yourself on your own.
Okay, and the question. So if Dr. Emily, if you're still on the call, or if she's not, anyone else who might be able to answer, clarify why the age limit has been set to 10 for the clinical trial that was just posted for nationwide children. And if there is this 10-year-old age limit, what is the criteria for compassionate care? Dr. De Los Reyes, are you still on the call with us? Dr. De Los Reyes, are you still there? We see her still on. Is there any anybody else? Oh, yes. Is it Dr. Reyes? De Los Reyes?
Hello.
Yes,
Yeah,
can you hear us?
Can you hear me? Yes.
Thank you, yes.
So, it is a very strict criteria with a 10-year age limit. So this is the first in-human phase 1/2 trial. So what that means is primarily it is a safety study to make sure that the children don't tolerate this intrathecal administration. Our experience at Nationwide Children's, and so when we make these decisions for inclusion criteria, it's not just me, it's a whole team of physicians and gene therapy experts. So the main experience is in very young children. So that that's probably the first reason why we set the limit at the age of 10, especially because of our experience and the fact that I think in children who are younger and healthier, that probably we will have less of side effects, if you will, or adverse events. So obviously this is the start of hopefully something new, and I'm quite excited that there's other studies that will probably go on in parallel with our current studies to look at these at the various approaches for the treatment of CLN3.
Thank you. We have another question. This may be for Dr. Meyer, but perhaps someone else as well. Can you please give a quick overview of what small molecule therapy is? I've not heard of it, but see that a few groups are working on that for CLN3. So actually, that may not just be Dr. Meyer. Anyone who's able to answer that?
I mean, I can answer it, probably, what it means for me.
Okay, great. Thank you.
So, obviously I'm a gene therapist, and I'm a really big fan of gene therapy, and there's a lot of things gene therapy can do. But at the same time, there could be small molecules, chemicals, maybe drugs that have been approved for other diseases that could be helpful in Batten disease as well, and maybe even in several types of Batten disease. We're talking about, molecules that maybe have been developed for neuroprotection, for synaptic support, and we can screen these kind of molecules, which we also have another collaboration going on, and actually Sanford research is very strong in screening these molecules, trying to find either molecules that could, be used in combination with gene therapy, or could be alternatives, or could, be used prior to gene therapy. So we're talking about really looking at drug libraries, working together with industry or any researchers that find molecules, any chemicals that could be used as a potential therapeutic agent.
Okay, thanks. I had another question come up, maybe beyond Batten or BDSRA, is wondering what upcoming trials or opportunities there are for those over 18 living with CLN2. Also wondering where Canada stands in all this, because I feel like we are so behind. If anyone's able to answer these online, able to answer and comfortable answering the questions, that's great. If not, we can work on getting answers to these afterwards. I recognize some of them may require some more information than able to be answered on a call.
Hi, this is Noreen with BDSRA.
Thanks, Noreen.
I guess part of my answer is that we will have to find some more information specifically about where Canada stands. A lot of times it depends on the pharmaceutical company who's working, where they want to locate their testing sites, whether that's in the U.S. and Europe and Asia and Canada. They make that decision. As for those living over 18 with CLN2, that also depends on inclusion and exclusion criteria, and we can sort of look through the ones that are posted, and we can look at what's open. Another option is, of course, as was mentioned earlier, compassionate use. I know for the Beyond Batten CLN3 project, theirs is open to compassionate use. Correct, Mary Beth, for over 18 soon, hopefully. I guess it's a case by case, but I'm happy if you want to, I think that was Monica. Monica, if you want to give me a call and we can sort of walk through each one of them.
Thank you for that answer. I think we hit all the questions that are pending. There was one question about compassionate use for Amicus CLN3 trial opened up December 10th, 2018. What is the policy? If Amicus, if you're able to answer that, or I'm not sure if that was part of Noreen's answer as well.
Well, this is Jane, and I'm here. So I just wanted to clarify that the CLN3 clinical trial that was just posted on clinicaltrials.gov, actually Monday evening, it got posted, is for clinical trial. It is not for compassionate or expanded access. It's a specific clinical trial. So I just wanted to clarify that. And that as far as the overall expanded access policy at Amicus, that is on our corporate website and can be found there, amicusrx.com. And our approach for Batten will be the same as our approach for other diseases, where a physician is able to make a request if they would like to access a drug, whether, an investigational drug or if a drug is unapproved in their area for a patient, and then it goes through a process with our heart team, which is actually handling expanded access review team. And our heart team looks at all of these requests and essentially determines how to best triage it. And I would say that one of the first things that's looked at is, are there clinical trials available for that particular indication? And if so, be they Amicus trials or other sponsored trials. And so that's one of the first things that's looked at. But I did want to just clarify that the CLN3 trial that was just posted on clinicaltrials.gov is a regular clinical trial. Hope that helps.
Thank you. Thank you. We have one other question that's come through so far. I'm going to ask this, and then due to timing, if there's any additional questions that come through, we'll direct them to the panelists and get answers out. So the final question is, what are possible adverse events anticipated for the CLN3 gene transfer study? And again, I'm not sure if anyone on the call is prepared to answer that right now. If you are, great. If not, we can get the individual the answer afterwards. I may be more thorough than on the call.
I guess I can answer this in general, what we've seen in gene therapy trials before, and also has been published and is well known, is that there can be a transient elevation of liver enzymes because of the immune system sees the gene therapy as a viral infection. And therefore you can have activation, obviously, of B cells and T cells producing antibodies, and you can get transient elevation of liver enzymes that have been extremely mild and non-clinical and have resolved in all cases. So these are the effects or side effects that we would expect, related to immune reactions, but none of them have been causing any troubles, and they were all non-clinical.
Thank you so much. Thank you so much. So in the interest of time, I just want to wrap up the webinar by thanking you all for participating and for all of our fantastic panelists. Global Genes, as I mentioned at the beginning, does exist as a nonprofit organization to help connect, empower, and inspire the rare disease community. We have a number of educational resources and events that are available to everyone. We have toolkits in a variety of different topics that can help you navigate your rare disease needs that are available on our website, globalgenes.org. There are other recorded rare webinars as well that are both disease-specific and non-disease-specific available on the website. And we have Rare University with a fantastic course for free online that's focused on the drug development process. It is incredibly educational. If you're interested in learning more about the drug development process, how clinical trials differ for rare disease than other disease, I highly recommend that you go to our Rare University, and check it out. And we will be launching within the first quarter of 2019 a genetics for rare disease course as well. That is incredibly thorough and it will be very beneficial. We have some upcoming events for next year. Our Rare on the Road Rare Disease Leadership Tour in partnership with Every Life Foundation will be at 4 locations this year: Boston, Sioux Falls, Birmingham, and Denver. Our dates are March 30th in Boston and May 4th in Birmingham, but didn't show up on my slide. In Birmingham, May 18th in Denver, and July 13th in Sioux Falls. And our Rare Drug Development Symposium held in partnership with UPenn Orphan Penn Medicine Orphan Disease Center is in Philadelphia on June 7th. Most of these are available at either no or very low cost, and I strongly recommend, if you'd like to learn more about those, to contact us and put them on your calendar. And with that, we thank you so much for your time today, and we will be following up and making these slides and the recorded presentation available for your review later, or to share that with additional individuals who may be interested. Thank you so much to everyone.